Let's start it. She said that she's coming. Good afternoon, everyone. Before we kick off the Win Distinguished Lecture, I'd like to mention the University of Waterloo acknowledges that much of our work takes place on the traditional territory of the neutral Anishinaabe and Haudenosaunee people. Our main campus is situated in the Haldeman Tract, the land granted to the Six Nations that includes the 6 miles on east side of the Grand River. Our active work towards reconciliation takes place across our campuses through research, like learning, teaching, lectures, seminar series like these ones, and community building, and is coordinated within the Office of Indigenous Relations. We are so fortunate to have here Professor Han Chen. She's coming all the way from Hong Kong PolyU. Professor Chen has a very illustrious career. She is a chair professor of molecular medicine at the City U of Hong Kong, and she's one of the highly regarded top cited scientists in toxicology, and has won a number of medals and honors. And also, she is very much committed to the public service, being the chair of the board of Nano and Advanced Material Institute, NAMI, with whom Win is planning to have much broader relationship. And Dennis was previously at NAMI as well, so it's kind of very quite coincident. So we are looking forward to Professor Chen's talk here, and welcome to Waterloo and welcome to Win as well.
Thank you. So thank you for the kind introduction. This is my first time visiting the University of Waterloo, and I did my postdoc at U of T. So this is actually a very good opportunity. And that my name is Shuk, and then the Cheng here, it is a Cantonese, pronunciation, because some of the papers you will see Cheng, and that is actually, as the first author, that is my PhD student. So I just want to make sure that, I will remember that. And that, so today's talk is actually much more about putting things in perspective. And I'm not talking to you about the latest papers and publications. I think that you can do all of that yourself. But I want to use, the vintage point of me, starting so early in life. And then at that time, there was not a lot of requests about impact and all that. But with this lecture, I'm going to show you an example about how things started. And then, totally out of my control, that I found that I've been cited, my work has been cited in a law journal. And that is actually how I started, to figure out how things are. So I know that in 2005, quite a lot of you probably were not born, but for me, that is, how I get started with nanomaterials. So this is, in New Orleans in 2005, and then I went to the Society of Toxicology conference there at the New Orleans. So at that time, why I went there was that I have always been, fascinated by embryonic development. Think about it, is that all of us sitting here, but all of us started with just one cell. How come, we have the hair? How come we have the space between our fingers? That is essentially the most interesting thing on earth for me when I was, in that stage. And then, it's one thing to look at the developmental biology of embryos, but then what can you do, with all of that? So as an enterprising young researcher, I felt that I can set up the system to look at how man-made chemicals, even pollutants, and then drugs can, be seen as whether they get into the embryonic body. And since it's totally transparent, I'll show you some of the pictures, then, I can actually track it. So I actually have a lot of fun doing bioimaging, but then this is about the history. So when I went there in 2005, and then and at that time, all the nanomaterials were actually being talked about. And they actually have one session in this particular conference to talk about, oh, we know nothing about it. So, is it safe? Is it good for mankind, or are we producing anything that we will regret later? I mean, we have regretted a lot, like DDT or even microplastics. And so, so that was the general thing. But as a biologist, I knew nothing about it. So I learned about the physical and chemical properties, how the surface area is hugely amplified because of the small size, and then how difficult it was to characterize. And so, the whole thing after I walk out of that session is that we need to actually get the data on, whether this is something hazardous, and how each of us and everything on the planet Earth will respond to it when being exposed to it, and what is the mode of action. So that was in March of 2005. And then, I went back to Hong Kong, and then I applied for a conference grant, and I get a good sum of money. So just divide it by 6, and that is the equivalent of Canadian dollars. And so I got it from a local charity, the Culture Foundation, and then I used it to organize, this particular conference, Nanotoxicology, then environmental impacts of nanomaterials. And that was in May 2006. So this is, where it gets interesting. When you don't have enough ideas about what goes on, the best thing is to bring in the people who's working in the field to talk to you. And so, with the money, I was able to fly in people from US and France and also different places. And that's, where I started a collaboration with different, chemists. Now, this is the slide that I put in just to let you know what I have been working on. I know that you will know a lot more better. But what I'm trying to say here is that I started with carbon nanotubes, and then I branched out with gold nanoparticles because where my curiosity brought me. But also, I was one of many different labs in the world that worked on it. But to be one of the first people to actually ask a specific questions gave me, a lot of room to set up collaborations, because, at that time, it was before you can actually order carbon nanotubes from anywhere. It has to be made by the material scientists, and that is why the collaboration is important. And that was, like in the old days. So with this, in case you get lost with all my history talking, first of all, it's about the initial curiosity. I asked a simple question, is that if we're going to make tons of it, then, would that affect any of the biological system? And the brief outcome is that, as I say, I actually realized that one of the law, the journal, one of the papers cited my particular paper. That's get me started to look at it. But then, I also branch out into gold nanoparticles, because I play around with this, and then, as all scientists, if you play with one toy, you want to play with another one. And that, formed the basis. But what I'm trying to declare here is that I'm not the only person working on it, and it is, community effort, and the societal impact is not coming from my own work. I just want to declare it now, and then, share with you my journey. So with carbon nanotubes, basically they are rolled up graphene, and that all of the physical, parameters there. So I put it in here for one reason, because with the advance of AI, like, the very much materials research project in Stanford, they have the large language model for all the databases. And what I have learned from the people, developing it and using it is that the specifications are important, because you can go and ask any of these LLM about, I want, this to be this specification and that specifications, and they will, I'm not too sure how they work, but they manage to screen things through and then come up, with a particular chemicals that will, give you the kind of specifications that you want. So this is, where it becomes interesting. In the old days, I marked this down just because I have to publish it. But these days, I marked it down is that, okay, I can actually go and ask all the different LLMs that, what if I want to have like 10 times more the thermal conductivity, but, the 10 times less of the high tensile strength. So these days, you don't need to read 1,000 papers to find it. You can just go and search. And I think that this is, part of my education to learn how to do prompts. And that is why I share with you here. And for the same token is that in those days, these are the listed carbon nanotube properties, and that we have a comparison that we got. So what we have worked on are the ones that are being made at that time, and I'm sure that these days you can actually ask it for formulations and prompt it for like higher modulus or smaller diameters. And now, this is the fun part. In 2003 or the several years, there people were selling the dream of carbon, nanotubes as the nanomaterials. I remember I went to, one of those seminars, and they say that you can actually build a bridge from Earth to, the moon based on carbon nanotubes. And this is a history lesson to let you know is that if you want to sell something, sell something that is much more realistic, because you can get people to imagine, but most people will feel that it is such a letdown once they started, doing the experiments. So in the old days, there was no rules, and so they actually try everything, and then they talk about if they put it in tennis rackets, they can sell it for 10 times more, and actually, it never happened. And so that was, all of the hype, and that at that time they thought that, in 2005, by 2010, they would have a market that growth to billions. And then we have, the biologists, that look at it and say that, hey, this looks like, in the shape of the asbestos fiber. Are you sure that you want to release the carbon nanotubes? And would it, get the same thing if you want to put it in the ceiling and, some of it will flicker off and get breathed in? And so that is, the whole thing about, carbon nanotubes is unfortunately the shape is very much, like asbestos. So that is, how we started. Now this is a zebrafish embryos, and normally it will have a chorion, to hold it. That is why it looks like a curveball. And then, if you take away the chorion or after it hatch, it will look like this. And why am I interested in working with this since, I started my tenure work? It is because it shares a lot of the genes with humans, so it is known as a vertebrate, model. But also, if you're being sensitive about animal testing, the Europeans has got it sorted out. So because it cannot feed, so it is not an animal. It is food for other animals. So they actually, have it in their legislation to say that that this is not, animal testing if you use zebrafish embryos before they can feed. And that gives you actually 5 days, 5 full days to look at this, wonderfully, mobile and transparent animals. And that, that is why I got into it, because I want to have anything that I've tested to be able to go further into the market than just publishing a paper. And so I have to be mindful about the society and what they feel about it. Obviously, now we're in 2025, things are totally different. You have all the organoids in the world, so you can actually, reconstruct a brain and eye with different cell types. But then, as a biologist, I can tell you, you cannot replicate everything in, an organoid, all the organs on a chips. For example, immunity is impossible to do. Yeah, so that is one of it. But this is a picture to show you, like in the old days, this is, a very, very old paper, 2007. And then we could see that, when we exposed the embryos, we found that if you expose it to some single-walled carbon nanotubes for 42 hours, you get some pigments. So the animals, they were responding, and we didn't know, much about what happened. And then the other thing was that I show you. So when I exposed the animals, they were actually protected by the chorions, which, it is quite a good barrier. They will allow for gaseous exchange, but nothing else. So why are we, seeing that? My paper didn't, give you the answer, but we have shown that, the single wall carbon nanotubes, the exposure cause hatching delays. And then, but then the animals somehow catch up, and so all of them hatch. Now this is, the compare with, what Sushanta showed me about his microscopes. This is really, the scale of biology. And this is, the chorion and the membrane. If you look at it on the outside, you get to see all these dots. And then on the inside, these are nanopores. So that acts as a gaseous exchange. And in the old days, as a biologist, what I did was that I just add the carbon nanotubes into water. And we all know that it will just aggregate. So they are huge lumps that will not get past any of the nanopores. But, we actually saw that with carbon nanotubes, with increased, so this is a dose response curve, with increasing concentrations, we could see, an increase or a decreasing number of the embryos hatching. And so we, mark out two of the concentrations, and then we look at, more time points, because it's very difficult to do biology experiments, and you have to have enough repeats. So we actually have two, concentrations, and then we look at single wall carbon nanotubes, double wall carbon nanotubes, and carbon black. So what is carbon black? That is, your printer ink. And so at that juncture, I just want to say here with the carbon black here is that at that juncture, carbon black and carbon nanotubes were regarded as just carbon in the eyes of the US EPA, the Environmental Protection Agency. And so, so we show that, okay, they delayed, but most of them actually catch up later. So we actually, talk about is that if there might be the heavy metals being used in the manufacturing of carbon nanotubes that, came with it, and that is why they could cross the barrier of the chorion, the little membrane, and then delay in hatching. But the animals, they managed to catch up, and then we look at every single organs, about the hatching, the hatched embryos, and there was no developmental defects. So we only, found out about the delay in hatching, and we published it in Environmental Science and Technology, and then they marketed it by online news. In the old days, there was actually very advanced of them. Most of the journals don't do that. And then it got, picked up by, another online news. And so that was a 2007 paper. And then in 2010, we found out that, the US EPA actually come out with a significant new use rules. So that means that they come out and say that it is not equivalent to carbon black. So it is a significant new use. And then they have to, introduce rules to control it. And so that was, the first hit was that they published it in 2010, and then they cited 39 papers. So that is what I claim was that I'm just one of a large team. So mine is one of the 39 papers that they cited to justify why they have to put in a significant new use. Because the rules, once they set it as rules and publish it in the Federal Register of the US government, then, it will have impact. So in the first, the publication, they actually asked for a 90-day pre-manufacturing notice of new, carbon nanotubes. And then they have subsequent version that makes it, the filing of all the materials was becoming very, very difficult. And then, like all scientists, I want to play around with it. So, I put it into, different systems inside the carbon nanotubes. So I test, whether if you functionalize it, that is, that you use it and then just put. I got the idea because I have two young sons and they play with Lego. So you can imagine, my concept is that if we stick things to it, will it act differently? And at the old days, we really need to have the carbon nanotubes from our collaborators. So we have the multi-wall carbon nanotubes, we have the double wall carbon nanotubes, and also we play around with the different, length of the carbon nanotubes. So these are, a lot of this work was done by my student, Cheng. So that is, why I said that when you see Cheng as the first author, it is not me. And so, we play with it, and like all scientists, we want to tell the world around it. And so we actually went to this particular the invention exposition in 2009. And we went there because, for us, is that the Korean society is very conservative. And they actually have a Korean International Women's Invention Exposition. And so we actually pay the money and go there, and just to show, solidarity to other women scientists in the country. And we got, oh, sorry, yeah, I pressed the wrong button. And so we got, let me see, I'm doing something wrong? Too excited. Oh, oh. Okay, yes. So, but this is, another lesson I want to encourage all the students is that when you have something interesting, go to competitions. Obviously, it might be very good if you get the award, but to be able to stand up in front of, your booth and explain things, that makes you, have a good interactions. So that is another take-home message, is that don't care whether you get awards or not, but then, do that. And go, and go into competitions. And so, we have, been part of the machinery that raised the alarm about how to control it. And so the field is actually still grows. And they were projected, to be billions by 2010. And then now the projection is that by 2030, it will be, billions. And so that is, the difference between the hype when it first came out to the reality about generating GDP. And these are, instead of going to the moon, which they've never managed to get the grant to make the nano bridge, these are things that are being used. So if you look at it, there are a lot more of it is into things that people use to make it more durable, and while doing things cheaper. So it's still, it is a material being used by a lot of people. So after testing and playing around with all of it, Jinping, the other Cheng, my student, came into my lab and kind of looked very worried, because all her life, that in the previous few years is about showing carbon nanotubes are toxic. And then we come out, and find that it is not toxic. So she was really depressed because she will not be able to graduate. And then I say that, okay, if it's not toxic, then let's use the same, Lego, and see whether we can develop it into, therapeutics, because we have an in vivo model to test things. And so that was, what happened. And then I, collaborated with, Yapin. He's in Clemson University, and then we make, more Legos. So this time, we use the single wall carbon nanotubes, and then we just use the PEG. So these are the Lego bricks that you put in. But then we want it to be reversible, just to play around with it. And so at that time, we could say that it is low toxicity, the cells are doing quite well, and then it is reversible nuclear accumulation. That means that what you put into the cells, the carbon nanotubes can go inside and then go out again. And that will be, cause it will never be biodegradable. We have something that is not biodegradable. So the next best thing is that it will at least, get out of the cell. And so we managed to show that. And then we, do much more things is that, okay, if we think that it is safe, then the easiest thing to do is that look for an application. And that because we are using cell lines, so, one of it was that we want to look at whether it can be used as a drug carrier for overcoming multi-drug resistance. So multi-drug resistance, it is a phenomenon that, for the cancer cells, when you expose them to the same cancer drugs, they will become, less sensitized by it. And so that's the development of multi-drug resistance. So we think that if we have a good carrier, like the carbon nanotubes, would it be able to, fool the cells a bit, and so that it will have less chance of developing the multi-drug resistance. So we found that, it is a very effective drug delivery, and then we think that you can have more of the same drugs, and then, it will enhance the chemotherapy outcome. So these are all done in cells. And then we further asked, because it's like Lego, we play with like pH responsiveness, so that, we will learn more about what can be done. And then we, other than cancer drugs, because silverfish embryos is so transparent, you could actually see all of their blood vessels. So if we have a system where we could see all of the blood vessels in a vertebrate, animal, then we can track things. We can, see whether where it goes. But much more importantly is that the process of angiogenesis, it is something that, tumor cells do. For the tumor, it is small, but when it grows bigger, it will, get the blood vessels to go in and supply the nutrients and oxygen. And so one of the anti-cancer therapeutic strategy, it is to be angiogenic, to make sure that they don't draw in the blood supply so that, their survival will be less, successful. So, we played around with it, and then we found that, the same Lego principles can be used to attach anti-angiogenic, the drugs, and hopefully, that somebody else will pick it up. And that we also, want to know is that for the carbon nanotubes, they will interact with the proteins, of the tissue of the cells. Would that be any difference if we use different types of carbon nanotubes? So we actually, do the different types. And at that time, we use the multi-wall carbon nanotubes and carbon black. And then we asked the question is that do they bind to the same types of proteins? And we found out that they bind to distinct protein sets. So that means that if, we will have protein signatures to see whether it is safe or not, and that we don't need to use carbon black, or we shouldn't use carbon black as the baseline study. So that was what this paper was about. And then after playing with, carbon nanotubes for so many papers, we want to play with other materials. And so, we want to use nanoparticles. And this time, we want to coat it with polymer to actually do the charge-dependent cellular imaging, a bit like, a lot of the more modern imaging reagents. So that, it is our first step to move from, the tube-like nanotubes into nanoparticles. And then we, work with graphene oxides. This is another, toy that we work with. And then we work, switch to gold nanoparticles. So for this, I just want to show you the journey is that when we have a good system to test things, then we want to play with it by testing it with, different materials. And for the material scientists, I think it is important for you, to look at things is that if there are two things. One is that you have a good, measurement. Then you make new measure, the equipment, like what Susanta is doing, new microscopes. Or it is the same instrument, then you make different materials to get the data. Both of them are very important, especially at the AI stage, because all the large language model, they need data. And this sort of playing around, with different parameters will be good to give people directions. And so, at that time, you switched to gold nanoparticles because one of my collaborator, actually he's in PolyU, and then he's very good with the gold nanoparticles. And then we, use it because the gold nanoparticles, you can add a lot of different things. So that is, also my Lego like of, mind that we just stick different things to it to see whether we can increase the drug loading. Because all the drugs, especially chemotherapy drugs, they are poisons. So if you can actually kill the cancer cells with less dosage, that will be, very good. And that was the motivation why we do it. And that we, use the different method, and then with the functionalization, we try to, stick the different things onto the gold nanoparticles. And that not us, at that time, the whole field was going through it. And so these are the gold nanoparticles because they have the physical property and also they have the chemical properties. So, theoretically, you can actually attach radioactive therapies or photo, active therapies in addition to the chemical part. And this is, what other people have done, and I want to give them credit here because the field actually has moved on, not by me, but a much bigger community. And so, they are getting all of that, done. And then now I think that the hurdles is actually to get it into, the approval stage. Because like all drugs, the clinical trials are extremely expensive. And that is why what we do in the lab, we can have 300 papers or 30 papers. But at the end of the day is whether it gets into the clinical trials that will determine whether it will be useful. So the take-home message so far is that it's very easy to tell people that this is toxic, don't use it. But much, much harder to tell people is that I think this is useful, use it. Because, to prove it is not good, it's so easy. It only takes, 39 papers, and then all those carbon nanotube manufacturers will have to file like mad. But to say that, if we want to use it as drugs, then you try a lot more, and then you have to do the clinical trials. So here is for all of you to consider when you go into a direction, what do you want to do? Because to stop an industry is much easier than to develop an industry. And some of the brave ones here are getting into, drugs. And so I just want to, share with you. And that, so with gold nanoparticles, these are our own work, of what it can do. We really want to check whether it can do, not penetrate into the cells, but actually go into the nuclei of all the cells. Because then, you can do imaging techniques, or you can stick, the different things so that you can go for nanotherapeutics that will go all the way inside the nucleus. That is actually, still something very difficult to do because of, the receptor, the ligand, and the physical barrier. And so, that is what we have tried. And then the doxorubicin is a very common, cancer drugs that we, try to stick it with different type of functional carriers. So basically, we have shown, in principle, is that with the gold nanoparticles, we can enhance the uptake of doxorubicin, and then it is, 2 times more cytotoxic. So these are all in the cells. But imagine if, those poor patients who are suffering from all the side effects of chemotherapy, if you can actually cut the dose by half, whatever you are planning to do, it will be much better for the patients. So that was the motivation for us there. And then, the other thing which, we have started very early on is the multi-drug resistance. So that is something that we want to do is that if you can overcome the multi-drug resistance, first of all is that you do not need to keep switching the chemotherapy drugs, which is very important for the patients. But then you can also, reduce the chemotherapy dose together with the very unpleasant side effects. And so, we started early, and in the old days, it's like social media, the equivalent. Then, they actually let you know whether your paper is being viewed, so they have the top 25 hot paper. And so these are just, more and more of the different functionalization. And then we wanted to go for the precision medicine, because if we have good molecules, then we can go, it's like the targeted therapy. At the moment, a lot of it is being, developed using antibodies to do the targeted therapy. And then this is, an independent way of having the targeted delivery by using the gold nanoparticles. And that, so these are not my work. So this is, after all of that we have published, these are the peers that are doing different research and trial new things. So, and that there are, 30,000 publications using the gold nanoparticles alone in the past decade. So it is a very, vibrant field. And then they are going for, the clinical trials. So that, prove that our original concept about having a carrier that can be less toxic, more targeted, is being, used by many of my peers in the field, and they have gone much further, because they are now, going into the clinical translation. And then, so these are not my work. So this is, after all of that we have published, these are, the peers that are doing different research and trial new things. So, and that, there are, 30,000 publications using the gold nanoparticles alone in the past decade. So it is a very, vibrant field. And then we started early, and in the old days, it's like social media, the equivalent. Then, they are going for, the clinical trials. So basically, we have shown, in principle, is that with the gold nanoparticles, we can enhance the uptake of doxorubicin, And so, I just want to summarize what I have gone through here, that, I started something at 2005 when things were much, the more in person. You go to a conference, you hear what people say, and then you develop your ideas. Nowadays, you can have, virtual attendance. So what the take-home message is that actually, go and check out different types of talks and different conference, because you might need to branch out from your current project into something that is interesting and new. So in addition of asking, the OpenAI, you can actually do the search yourself, because you can get, ideas from all the different search, but your own field much better. So combine what you know to, what you do not know by the searching through the conference, then you get new ideas. And then, so this is, what I want to talk about, the curiosity-driven research. I think that at the end of the day, we are humans, and we would like to maintain that we can still do curiosity-driven research. But with the AI tools, it will cut down a lot of the time that, if somebody has done it and failed, then you don't need to do it. And also for the simulation, there are a lot of simulation databases out there. It will also cut down on the number of wet lab experiments that you want to do. The only thing that will not help you is that the failed experiments. If you look at all of the LLM, they are successful experiments. So no one will tell you that if you try this, it will fail, because the poor students in Brazil has tried it and it didn't work, but nobody published that. So that is the disadvantage about not publishing. But still, with the modern tools, you can actually cut down on the wet lab experiments by exploring the different things and stay curious. Yeah. And then, oh yeah, we were curious. So after asking about gold, we actually had a paper to, the nanosilver particles. We actually worked with the orthopedic surgeons to see, whether it promotes tendon repair. So I know that, compared with what I have done, your colleagues here have done a much, better way of approach. So that is, all about what I do as a scientist. And for the remaining slides, I want to give you a broader introduction to my institute and to the nanomaterials research in Hong Kong. So in CityU, we have the university level, the institute called Digital Medicine, because it is really combining with all the large language model, the digitalization. We feel that training a doctor now, we have to think about, that kind of tools that they will use. So CityU chose not to train the doctors, but to develop tools for the doctors in 10 years' time. And so, we have the Institute of Digital Medicine that we hope to be able to work with our partners, the institutes, to develop the clinical programs and train people, and also, make it more interesting for the students to study all of those materials. And then in CityU, we actually, focus on the pushing forward about health and life sciences. So we have a new College of Biomedicine that was only established at the beginning of this year, and then they put together the existing departments. So, what is so different about it is that this particular college, which I belong to, we have the biologists, the biomedical sciences. We also have the engineers. So the biomedical engineers are also in this college. And obviously, we have the Department of Neuroscience. They, in this age, they deserve a department of their own. And then we also have the Department of Biostatistics. So, hopefully by putting everybody under one college, we'll have more interdisciplinary research. And then other than that, we have the only veterinary college in Hong Kong, and that the program is being accredited. So they always say that between London and Melbourne, we have only one, now we have two, the Koreans got it. So only two vet schools got the accreditation. And that's, huge investment from the university. And then, so this is like, all the propaganda slides, just to tell you that, our College of Biomedicine, it has, a lot of very good people. But then, even for us, it is, ranked 16th for the nanoscience and nanotechnology. And so we have a very, good group of talented biomedical engineers. And now, I want to, devote the last 2-3 minutes to talk about NAMI. So it is actually called Nano and Advanced Materials Institute, and we just short term it as NAMI. It is the government-funded R&D center by the government, and so everybody there was on project grants, but the projects are competitive. And so it was set up in 2006 with only 10 staff, and by now we have 260 staff, 7 laboratories, and out of the 260 staff, 200 of them are researchers. Half of those researchers got PhD. So this is, where things are with us. And then, so this is, from the 2006 to 2025, the center has received 1.5 billion Hong Kong funding. So just divided by 5 or 6, then the Canadian dollar equivalent. And then over the years, they accumulate about $300 million worth of equipment. So the infrastructure is actually very, very, good. And then 250 granted patents. So in 2023, NAMI as an institute with only 200 researchers, it ranked the number 54 in the league table for non-profit organizations that has been awarded the U.S. patent. So I found out, that, and so it is just to let you know is that the patent application, it is also vibrant in Hong Kong. The only difference is that unlike here, the researchers don't own the IP. Here you own the IP, so you have a lot more leeway to play with things and also profit from it. And that the government really want Hong Kong to have more interaction with the Greater Bay Area. So it is not like the Tokyo Bay nor the San Francisco Bay. This is the Pearl River Bay Area. And also countries of the Belt and Road Initiative. So I think that this is the part that Hong Kong can complement the efforts here at University of Waterloo, because in Hong Kong it's mainly Chinese people, but in Canada you have different ethnic groups. So whatever you do, the cultural background, the potential usage will be different. But then in Hong Kong, we have that. And so this is also, to show you about, starting with materials is the slogan. Obviously, we go into the ESG, Internet of Things, renewable energy, 5G, and then the health, and also infrastructure. So these are the, out of the 250 patents and many more projects, the emphasis on. And that, so as a government-funded R&D center, is actually to serve the small and medium enterprises of Hong Kong. Those companies that cannot afford to have that $300 million worth of equipment. So it is really about the smaller enterprise. They are the jobs, generators of Hong Kong. And the core technology platforms are all here. I think that like wind, you have very similar sectors. I will not talk about it. But I want to, use this remaining time to talk about commercialization. So for NAMI, my role is that I have been a member of the board since 2010. Now I'm actually the chair of the board of directors. So in Hong Kong, it is really about the corporate governance. I am, with the board members, we oversee how the institute is doing. We hire the CEO, we give them a hard time, and then, so this is the kind of commercialization that we actually, after, all these years, we can boost about it. So I'm sorry about, the Chinese, because the concept, about how to stay cool in the summer, they have a word for it. But what it is that these are modular, integrated, the construction methods. So it is like all the boxes linked together. In Canada, you don't need that because you have all the space in the world. But in Hong Kong, these are, the expected. And then the government is under pressure to build many more of them and faster and at lower cost. And so this is, something that we developed with one of the construction company, Chevalier, and that they, help to have this sort of lightweight concrete. And also this is, an example. What you're seeing, the pictures here are actually from Shanghai. So NAMI has, a not very glamorous technology. It's about, filling, all the backfill of when you dig out, the roads to do repair, you have to backfill it. And, it actually used nanotechnology and to create materials that will be, very fast and safe. And also it used recycled construction. So I'm showing you here is that compared with wind, this is actually not glamorous at all, but it is everyday life. And it is, making the industry easier to go forward. And so you do not need to have, huge donors or go through clinical trials. It is very run-of-the-mill, but that is the kind of mission that the government wants NAMI to do. So just to show you that. And then when it comes to drug delivery, if you can see that was me over there. So this is China Resources Pharma. So the company was set up 80 years ago in Hong Kong, and then they were set up in Hong Kong. They're the China Resources. From the name, that that they are from the big Chinese money, and then they work with NAMI because they have a need to make bioavailable turmeric. So, we all know about turmeric or the anti-cancer. Obviously, you can keep eating curry. But there is a technique about, the encasing it so to increase the bioavailability. And this particular company, wanted to do contract research. So that is the MOU, and then we will sign the contract research later, and it will be about 1.2 million Canadians. And that is one of the single most, or biggest project for NAMI when it is not to do with constructions. With construction industry, all the contracts are very expensive because of the materials. And then this other one is something about the nanoswab. So we all know that you can just get some of your DNA cells, but then they make, those little prickly thing that will be easy to do, and it will collect the cells, from the patients. So it is really about collecting enough cells, but without causing the discomfort. And then this is, one of the schemes. Maybe you can ask Dennis, about it. It is, a scheme that the Hong Kong government does. It's called Public Sector Trial Scheme. So basically, you can get a research project funded by the government, and then once you completed it, you can go back to the government and get, more money. So it's one thing that you can do it in the lab, you can measure it, publish your paper, get your patent. But then, if you want to use it for field trials, you have to have the initial, field tests. And so the money is for the manufacturers to produce enough of it, and then you can put it out field testing for about 12 months. So this is, a very, with my experience, is that this is actually one of the critical factors to make the translation much more, easy. And that is what I'm trying to, I'm share with the colleagues here, is that, so this particular thing is that we have all been to the airports, we have all the trolleys. And for the airports is that they actually do not know where that, 5,000 trolleys are. They have the Internet of Things, but for Internet of Things, they have to have batteries that can last a long time. And that is the trouble. So with this, what NAMI did was that it helps out with the casing of the batteries, so that the batteries last longer, and then, the airport authority can track it. So it has gone through a public sector trial scheme, and then they tested it on 60 of those trolleys, left it out, and get lost in the whole of the airport, and they collected the data, and then the airport authority was so impressed with the data, because instead of changing, the batteries every 2 years, they can now change it every 5 years. And if you have 5,000 trolleys to do, this is a huge thing. And last but not the least is that we all know that, with computing, it generates heat. And so, this is what they have manufactured with the Nanotechnology, is to use coolant. So basically, they dip, the whole hard disk into the coolant, and so that it will, dissipate the heat. And that is being used by, the civil aviation in the control tower simulator. So these are, the difference. What I'm trying to say here is that if you develop something interesting, if the university, the provincial government, they're willing to test them, and better still is that they're willing to buy your product, it will make, your startup company being able to survive much longer. And this is what I'm trying to say is that in Hong Kong, we have tried out different things, but this is one of the very good schemes that you might want to consider here, knowing that, Waterloo is very, a most fertile place for startups to survive. And so this is, all of it. So what I want to end my lecture is to recap, is that when I started, I wanted to do something curiosity-driven, and then I worked, with different people, and some of it turns out to be useful. The others turn out to be a really long way from being able to apply. But then with, City University, the university have different emphasis. So the digital medicine or the new College of Biomedicine. But on top of that, I also serve, as a voluntary worker for the NAMI or as a board member. So I have witnessed, outside of a university, what has worked and what doesn't work. So for all of you, the aspiring entrepreneurs here, make sure that you find a way, not just, to have an idea or an innovation, but when it comes to impact, you have to have a product, and then you have to have your product being field tested. And better still is that you have to have somebody willing to buy your products. So think about how to, generate this ecosystem, at least within, your university, because you have so many startups. I'm sure that some other startups will want to buy your product as well. So you have a very fertile ground just to think about this way. And then if the provincial government is buying your product, then somebody in Brazil or in Malaysia will think about twice about saying no when you want to sell your product. So this ends, my talk. It is not strictly a scientific talk, but I'm sharing with you my decades of experience in nanotechnology research. Thank you for your attention.
Very inspiring talk. Open for questions, comments.
Professor, thank you. I was going to ask about NAMI's perspective on bringing outside intellectual property into the work. Is there, like you mentioned, there's a public sector translation scheme?
Mm-hmm.
Would that be just for technologies developed in NAMI by NAMI researchers or adjacent, or is it?
Yes, that actually is a very good question. So the scheme actually is open to all Hong Kong, that we have the Innovation and Technology Fund, which is a grant agents that fund that. They not only fund NAMI, they fund, the professors, they fund the SME small companies. And so I'm not too sure whether if you have a Canadian startup, you can apply for it, but we can explore, because Dennis actually came through the system. So, we can ask him to explore. But the scheme, whether you get it from Hong Kong or you get it from your local, more local sources here. But I think it is really worthwhile to have a scheme where you can generate enough numbers of prototypes to do the field testing. And with NAMI, it is, it is a government agency. So you know, they do enter, actually a lot of their projects are what we call the sponsors. So if your company is willing to put up 51% of the cost of a project, the company owns the IP. And that, you know, that they actually give you 400% of tax rebate on the money that you've gone into. So you can see that Hong Kong is desperate about, you know, trying to get people to do innovation. But I think that if you have some good ideas, think about, you know, doing the field trial. From what I have shown you is that it makes a lot of difference if you have the field tested data, even for 60, you know, of them over 6 months. And then for the backfill, you know, if you have something interesting, there are different companies that want to test it with you. So I show you the Shanghai example, and some other technology we will be, you know, field testing it in Malaysia. So the take-home message is that if you have a good idea, field test it, and then reach out all over the world. I think that good products come from this sort of, you know, cycle, but the field testing part, it is important.
Thank you.
Hi, I'm a yeast cell biologist, so I have a biology question, and it
Sure.
was mainly with your zebrafish. The fact that you got early on, I think at 57 days,
Hours,
hours, that you had
a delayed in hatching.
yeah, and then but then by the end, by the 75, you had complete recovery. Did you ever figure out what the mechanism was for that? Because it's the converse of what we usually see when we see in toxicology.
Yes. So for that, you know, we really try to figure out how come that they managed to catch up, okay? And so far, we still haven't found it. So, you know, for us, we just put it down to the robustness of the embryos because they, you know, in addition to what is distracting them, like the toxic, you know, the heavy metals, they have, you know, they have their cell-to-cell signals that, okay, you know, we are forming the heart. I mean, where are the, you know, blood vessels, you know? So I think that it is the intrinsic cellular signals that tells them is that, okay, you know, you have to be there, a particular cell type. So that we think is about the catching up because we didn't, you know, grind them up and see whether, you know, their increased expression of the genes. So that was, you know, not being done.
Well, I was going to say we couldn't do qPCR back in 2006, right? So, but if you went back now, that would be because it would be really interesting to know what got upregulated to allow that to happen because it's potentially really powerful.
Yes, exactly. So hopefully, you know, some of the young researchers here pick up the cue is that is one thing to accelerate it, and is another to, you know, make sure that the legacy, the delayed process can be, you know, brought back. Not you know, because I show you all the embryos, they ended up very normal and happy. That means that the catch-up mechanism was so well tuned that they did not overshoot. Exactly, you know, yeah, we appreciate that it could be powerful. It's just that in 2007, no single cell PCR, no.
Thank you for your interesting talk. So in your comment about the nano aspects, because many companies are avoiding using nano, like Europe need additional verification. So what are your views on nano in regards to commercialization? Right? Because many companies are
Yes,
like cosmetic company, for example, right? They will not put nano as a selling point in their product, even though
they
nano
are
is
using
an
it.
important enabler to their performance.
Yes. So I think that for you know, for all the nanomaterials that are integrated, like, you know, in the soles of your shoes, then they do not pose any risk. And it is, you know, that those things that are being used that it can be washed out, like all the personal care products. We now know that a lot of our face creams got microplastics, belatedly and, you know, regrettably. And so I think that the regulation side is always playing a catch-up game. And that is why I cite this is that very rare that, with the papers published in around 2007 and all my other peers, that 2010, only 3 years later, the US EPA came out and do something. And for a government, I thought that is lightning speed. That is why, I chose it as an example. But my viewpoint is that with all the chemical engineers, hopefully you're not going to say something, is that it is a catch-up game, yeah.
Just to follow up on this, you mentioned, gold nanoparticles and things like that. But now there's a lot of new kind of 2D materials like hexagonal boron nitride, molybdenum disulfide and things. So do you have any things about how these uptick can take place in this kind of embryonic cell zebrafish and others?
Well, first of all is that zebrafish embryo is not fashionable, now. So that is why everybody's gone on to do the organelles, organoids, and organs on their chips or multiple organs on their chips. So I think that with what you're asking is that they can do, certain testing, either on the efficacy of it or the toxicity of it, but it depends on the choice of the testing materials. And anything, that have any remote chances of being used on humans, the road to the phase 1, phase 2, and phase 3 clinical trials is so expensive. And now these days with the the political thing is that if you being tested by the FDA in the US, it will not be recognized, on the other side of the pond. So if you have a drug, you have to go through two different types of clinical trials, and I think that is actually very worrying. But as things are, if the drugs are being made faster and cheaper and also safer, then we don't care where it is being tested. But whether you can buy it when you need it is another matter.
My question, thank you for your talk. My question is about commercialization.
Mhm.
So, do you see any trends as to which technology gets the most traction and who initializes um, the project? So, does it come from NAMI or does it come from the clients? Do they come to you with different problems, or do you seek out where you can apply your technology?
Right. So NAMI doesn't represent the whole Hong Kong ecosystem, but it is something that I know. So, in NAMI, what they have is that they have what we call the seed platform. So the researchers think that, okay, we know that the heat, dissipation, it is important for all of those power banks. And so they develop something like that. But because, it's government funding, so we actually have what we call the business development team to go out and talk to people. So, the typical question is that some companies say that, we have been a agent. We are selling paint, that is you can use it on your roof so that it will leak, much less. Okay, so the company will come up and say that, oh, we are the agent to sell some German products that can, do the job, but very expensive. And also we are agent to sell some, Vietnam mainland made products that is like 10% of the cost, but didn't work that well. Can you come up with something that we can, have it our own brand and sell it at half of the price, but do it as good as the Germans? So this is the type of interaction. And so that is why the business development group is there to actually talk to the client. Okay, if you want to do that, what do you want to do? And then we obviously have the technical team to talk about the specifications. So what type of specifications? And for commercialization, in the Hong Kong system, they have, the vanilla type of curiosity-driven, grants, and also even from the Innovation and Technology Commission. So you can just get it to do a proof of concept. And then they have, the public sector trial is that you want to make enough prototypes of it. And obviously there are, companies. So to approach the companies, you need the business development team. And that is why I'm making suggestion is that there are, you have such a, good concentration of startups. They all have very good ideas, but, to approach companies that might want to co-develop with you so that it will become a product that they can sell takes a lot more, of the efforts. So with Waterloo being so famous of your, internship program that has taken like 70 years to develop, I'm sure that this will be developed much faster. So to have an ecosystem where, things can be made in small batches, and then you can have the, iterations to test it. But actually, willing to test it is really important. And it is not something that can be done, in this room. But I think that the powers, out there, if they actually put in their efforts to persuade, maybe the university will want to try out your product, this flashy, water bottle, at least you get the field data. Yeah. And if they can fund, making enough of those water bottles, that would be even better. And the next step, which actually is very important, I didn't say, is that if the government procurement adds, that particular specifications into what they are looking for, then the company will have an edge. And your company or your ideas will survive and thrive. So this is what I have learned, in the ecosystem of Hong Kong. Somebody has to field test it, and somebody, in the government for the procurement. Because at the end of the day, ordinary customers like us will buy one water bottle. But if the government is saying that, oh, we need to buy, 1,000 bottles for our policemen, in their gym, and then they have the procurement process. And all procurement process will have specifications. And that is where, the focus or the efforts should actually spend on now. Think about it, that whatever you're thinking there, if you can go and say that the Ontario government ordered 1,000 of, this water bottle that's got my little paint on it, then you can sell it in Malaysia or Brazil straight away.
Maybe let's wrap up and